So welcome everyone. Great to see some familiar faces as well as quite a few new ones. As you probably know from the webinar invite, we're here today with our co-founder Nathan Clark and our client solutions engineer, Nathan Ballou. We'll kind of call them Nathan Squared almost, our power duo. So they'll be discussing some of the problems with lab data management in biotech today. They'll also walk you through our approach to lab data and demo our platform. A couple of things I wanted to point out before we begin. To try and make this webinar a bit more fun and interactive, we'll be sharing a few polls throughout, including an icebreaker of sorts that you can find now, and the polls button on the bottom right of your screen. Also, I encourage you to submit questions using the questions button right next to it, also on the bottom right of your screen, sort of throughout the presentation. I'll do my best to answer sort of simple yes/no questions live, but for ones that require a bit more of a lengthy answer from either of the two Nathans, I'll bring them up in the Q&A section at the end of the webinar. So with that said, we could probably turn to introductions. I'm Alan Kramiak. You may have seen my face in some of the webinar invite emails. I'm the founding scientist and senior business manager here at Ganymede. I'm a wet lab scientist by training, received my PhD in biomedical engineering at Columbia University, and I've got about 10 plus years of experience in R&D, mostly in preclinical drug development within oncology. My role here is to really inform the development of the platform, making sure that the data flows accurately capture the underlying scientific workflows, and I also work on developing collaborations and partnerships. And so I'll pass it on to Nathan Clark.
Good morning, everyone. I'm Nathan Clark, the co-founder and CEO of Ganymede. Prior to this, I worked at Benchling in product management on some of their data products in machine learning and business intelligence tools. And then prior to that, I have a similar background in data and machine learning systems and financial technology. So very excited to present here today and do our first webinar, but I'll also turn over to Nathan Ballou.
Hi, everyone. I'm Nathan Ballou. I'm a client solutions engineer here at Ganymede, so mostly work on implementations, doing demos, that sort of thing. My background's mostly in data science and analytics, and I'm really excited to talk to everyone here and show off Ganymede.
All right. Let me go ahead and share our presentation for today. So just to kick off this, good morning, good afternoon, and good evening to everyone wherever you are in the world. We'll start here talking a little bit about the philosophy of data in the life sciences from Ganymede's perspective. Life sciences, manufacturing, really anywhere where there's complex physical processes going on that have an analytical component, but nowhere is more complex than biotech and pharmaceuticals R&D. And so we do focus a lot on that, and that's our bread and butter. So Ganymede in many ways is inspired by looking at some of the top tech bios out there, the top pharmaceutical companies that have made major investments in their IT and software systems, and seeing what they've built over time. And what's striking, I think, to me and everyone on the team at Ganymede is that every single time they rebuild the exact same infrastructure stack, it's the same set of integration capabilities to lab instruments and to apps and LIMS systems, the same data lake architecture, the same set of computational pipelines. And so the inspiration for Ganymede in many ways is to offer that out of the box and try to bring that from those top companies, enhance it for them, and then also bring it to earlier stage companies or companies that haven't been able to make that investment in software engineering. You know, in 2023, you should not have to be building out cloud infrastructure. You should not have to worry about AWS or how you set up your data lake. These things should just work, and that's what Ganymede is all about. But to set the stage a little bit for how we think about the world, you know, this is, again, a sophisticated group, so I'm sure this is all stuff that you're well aware of, but I think it helps to contextualize how we think. There's a few phases of how data management has gone in the life sciences and generally in things like manufacturing as well, but especially, again, biotech R&D is one of the most complex cases out there of anything. Pen and paper dominated the world for a long, long time and still does in many cases. There's plenty of places that are still using paper batch records for manufacturing, paper lab notebooks for early stage R&D. And That's not to blame anyone doing that. There's a good reason why that is. It's because it's highly flexible. And electronic systems, especially in the old days, mainframes, brittle on-premise setups, are very inflexible and don't keep up with the pace at which R&D moves and at which physical processes evolve. And so inevitably you have a huge amount of human in the loop input and you're capturing data, but really humans in their brains are the ones maintaining the knowledge and maintaining the synthesis of everything going on. And the notes are really just a record for them to try and keep track in their heads. And so from our perspective, what has really enabled biotech and R&D to start moving into the 21st century, I would say it's significantly behind other fields. And again, that's not the fault of anyone in biotech. It's the nature of the complexity in the space. And so from our perspective, it's taken a lot of evolution in cloud platforms and tooling to get to the point where there's enough flexibility and ease of use to actually justify trying to structure and bring this data online. And so in the early days, you had some of the earliest ELIN and LIMS systems, things like IDBS, older MES systems like master control, for instance, that started to electronify mostly in an on-premise based sense. But really what's caused the revolution, I would say, in the 2010s and early 2020s for electronic data capture is the fact that tools have become cloud-based and they are flexible enough in how they're designed that you can have input from the end users and the subject matter experts in how the data capture is structured. It's not having to go talk to software engineers in some central IT team to say, hey, can you please update this web form so that I can capture new data? Sometimes that still happens. But things like Benchling, for instance, on the ELIN and LIMS side, or Printis on the MES side, both of which we see as leaders in this space, have grown tremendously because they get humans in the loop on structuring their data. And that informs how we think about the next phase of all of this as well. This is Industry 4.0, I would say, is a tremendous buzzword, but it's a fairly valuable one and a good frame of reference for us, where we look at, so Industry 4.0, if you haven't heard the term before, describes the evolution from original industrial revolution, things like mechanization, steam power, onto 2.0 of bringing electronics into the lab in the early 1900s and into the manufacturing plant in the early 1900s. Industry 3.0 came when computing came along. Now you have mainframes, now you have potentially personal computers, and the processes are digitized. They're not just analog electronics anymore. And there's programs that capture data and run analysis. And this is already a big improvement. But still, those programs are siloed. They're in physical computers. The hardware and the physical location of things is conflated with the logic and the software that's running. In Industry 4.0, there's a lot of promise in it. People talk about things like AI and machine learning. People talk about things like augmented reality, autonomous robotics, et cetera. But fundamentally, what I think is the most meaningful part of it is the fact that the software and the hardware are decoupled. The software and the business logic of the process moves into the cloud. And to enable that, you need data, you need connectivity, and you need everything to be in an integrated format. And that's the soul of all of this. I can say is having built multiple machine learning teams across different industries, machine learning is very hard in biotech, not because intrinsically the stuff is hard to model. It's because, especially when it comes to wet lab processes, it's very hard to have the data in a clean and usable format at all. Data is everything here. And so to really enable some of these really sci-fi technologies, we have a lot to do first on the data side. And I think that's what you see when you talk to IT leaders and experts in the space and software engineering leaders in the space, how they're thinking about how the future of their company's data is it's very data focused, it's very cloud focused. Things like AI and machine learning or IoT or AR and VR, those come after, and those are great, but they're blocked. Again, biotech is once more blocked relative to other industries because of the complexity and because that inhibits data capture. And so this is everything to us. People are looking to find a single good home for all of their data and break down silos between their data and have a single place where you can go see all your data and touch it and make it interoperable. Use that data as a control plane for the physical world, being able to say, I can run analysis, I can make decisions and implement them automatically in the physical world off of this data. And to enable all this, everything has to be cloud-based. So the data all has to be in the cloud. All of the interfaces to controlling instruments or APIs to apps, those all have to be in the cloud. And only then can you actually start to even talk about things like data automation, process modeling, maybe some basic machine learning and simulation. That is all downstream of that. So with that, I'll pause. Alan has a poll for us, if you want to issue that, Alan. So the poll is, what data integration or integration is most key to you? We see a lot of different areas in the life sciences and in manufacturing that are trouble pain points for people. Everyone has a different view on this, but categorically, these are some of the biggest ones. So we'll hold this open for a minute, and we'd love to see what people's reactions are in terms of where you see the most pain in your day-to-day or in your company. All right, Alan, I don't know if you want to read out the top results.
Sure. Let me see if I can access it on my side.
Bear with us, everyone. You're seeing our first webinar in action.
Yeah, maybe I'll give me a second. I'll try to figure this out on my end, and we'll come up with the results closer to the end in the Q&A.
Okay, great. That sounds good. Let me go back then to the second half of talking a little bit about Ganymede before we show you a demo. So with that, all this philosophy leads into how we think about Ganymede's approach to the world. We want to take this infrastructure that we've seen developing and make it ubiquitous, make it easy to use, and make it easy for you to develop on top of with your engineering resources, or for us to develop on top of as services for you. Fundamentally, the types of problems that we see ourselves solving are threefold. And this describes generally the flow of data in the lab. First, you have to integrate something. You have to be able to get the data where it's at from oftentimes lab instruments, maybe from PCs or sources on the network, maybe from databases, et cetera. Our tools are meant to integrate anything. We have a variety of agents that we can talk about. We have the ability to build your own parsers. We have a huge library of existing integrations. And we built this as the infrastructure to build integrations rapidly to your data, wherever it is. And once we have that access to the data, we will save it in Ganymede, where it can be pushed to your data lake or pushed into automation. The second phase is analyzing the data. The data is not very useful if you're just saving it. We've done a ton of interviews of people who have built out systems like this. And what's a really common theme is they did start saving all the data, but no one looked at it after a year. If you can't make something of the data from a semantic standpoint, it's not going to be useful for people. The data, just having an archive of data is not nearly as useful as doing something immediately with the data and automating things. And so that's where putting the ability to run arbitrary code that your engineers define, our no-code analyses or transformations, our ability to connect data from A to B and remap data, that's what we specialize in the middle of our cloud, which is the ability to, we are a full cloud pipeline system with the ability to run any arbitrary code and distill that down into certain no-code analyses and transformations that we have out of the box. And you'll see that today in our demo. And then finally, once you have that result and you have something that's meaningful to you, what do you do with it? Maybe you need to put it into an ELN or LIMS to change your lab for what your scientists see or record the data. Maybe you need to drive some automation off of that. Maybe you just want to save it into your data lake. Either way, our integration capability also is very good at writing to data and writing to any sort of structured data source. And so this at a high level is how our architecture looks. We call our architecture lab as code. And the philosophy behind lab as code is saying what's made other industries tame complexity is saying that you will define all the structure of what you're doing in software, in code, whether that's code represented as no code steps or it's true code that an engineer is writing at the end of the day, you need to describe what your process and your connections and your data flows are in software. And so what we do is when you have instruments, for instance, or really any data source, those will drop local files onto a PC or other network drives, maybe. Our agents are a variety of technologies that can meet the data where it is and sync it up to the cloud. And we always sync the data up raw. Only in the cloud does any parsing, any integration happen. We'll parse the data into our standardized tabular format. We'll run analysis. We'll save results. We can sync this in and out of your ELN and LIMS. And in fact, this is critical because having the connection to the ELN or LIMS, or at the manufacturing scale, your MES system, that lets us associate all of this against a common sample ID, a common batch ID, whatever your unit of account for your data is, to again, make your data more meaningful and enriched. And we save all of this into a data lake. It can be ours, it can be yours, where we can then apply additional schemas and ontologies into the data lake to give you additional structure for your data. And this view is a representation of how we think about the world. So Lab as Code is designed to say, once something's integrated into Ganymede, whatever it is, whether in this case, it's a bioreactor itself, represented as, say, files or an OPC connection, whether it's a probe stuck in the bioreactor, maybe streaming data over a physical connection. We have an IoT device for that. Whether it's something like LabVantage or Benchling or a thermal sample manager as a LIMS system, for instance. All of these things, once integrated into Ganymede, are represented as these tables, these data frames. And this, from there, makes it easy to reason about your lab automation. You can write code, you can use no-code connectors, you can write SQL to move data around between these and treat your lab, treat your actual physical lab and its operations like a bioinformatics pipeline. This is our secret goal, ultimately, is to say what's happened with things like genomics and higher scale informatics pipelines, where you do have this discipline of people going in and being data scientists and writing code to drive data around. That's been a transformation for the industry in the dry lab. We need to bring that to the wet lab and take those people and give them the tools to focus on the business logic of what you need to do with your data, moving it around, saving it, actually changing the internal state of the data. In this case, maybe taking your bioreactor data, taking your probe data, cleaning it up, and then putting them together and just adding them into LabVantage. It's just adding a record onto this table in Ganymede. We try to make that as easy as possible so you can purely focus on the business logic and invite those people in to self-serve, help define their workflows. And it's a tremendously powerful paradigm that's made us grow very quickly so far. One more view of this, and I know this is a busy slide, but we want to emphasize the way that we see the world is you may have the lab instrument out there, or again, any data source emitting some data. Our agents will collect it and put it in the cloud, and then we save every single thing we ever see. Ganymede's very sticky. Any file we ever see, any data point we ever receive, it's all saved forever. And then we'll process it down into our tabular data format where through those drivers, but these drivers are all things that are editable by you. You can make your own integrations, you can use ours or extend them, and we'll maintain the relation between the parse data and the original file so you have traceability. And indeed, the same thing is true when you do a scientific analysis or transformation from this process data down to an analyzed result. That's also user-definable code. You can use our out-of-the-box connectors. And again, traceability is maintained back from this end result to where the data came from. And so when we put something into an end destination, like a LIMS system or an app, you have the traceability all the way back to the original data, everything's saved forever. And we will store all of this, every single step, the file, the intermediate data, the end state data in our data lake, which can be synced to yours. And so you can have a complete copy of everything here. All of this code that we're running is, again, Ganymede's built as a true first-class cloud infrastructure system. So although you may never even touch the code, you'll know that we are creating a GitHub repo for you on the back end that's storing your code. So it's properly versioned, it's accessible. You can always tell what code ran what data at any time. And so compliance is perfected substantially more so than building this yourself. And in terms of where you can think about this as a result in your stack is we find that there's a good midpoint where there's a lot of raw cloud tools out there to build your own systems. People oftentimes will default to this. we really want to intercept anytime people feel like they have to go build something themselves in one of the cloud providers and find that daunting or find that to be a big investment that doesn't make sense. We have these tools out of the box ready to go on the cloud side. And then we also have the connectors ready to go to all of the apps and workflow tools out there, to all the instruments out there. We have a tremendous library of existing connectors to instruments and apps and analysis systems and so on. So our aim is to be the complete cloud infrastructure in a box for you. And to reiterate, just before we jump into the demo, the way that we see this is we need to give power tools to the people who can code and the data scientists here, whether that's our people, our solutions engineers doing services work for you, or your internal engineers using the Ganymede platform. They need to focus on the business logic and be expressive and powerful in there. They shouldn't have to worry about infrastructure or integrations. And that's what we fill in the gap on. And what they build will then be, they can generate no-code UIs to utilize the automation for scientists or research assistants or technicians. We also have a layer for that, or we can just integrate with whatever surface they're using, like an ELN or an MES, to automatically execute automation when they change something in those tools. And then finally, as we've discussed, we save the data forever. Compliance should be something that is natural and is automatic and doesn't require significantly restructuring your process for. So just to do a final review here, Lab as Code connects everything. Anything we can get our hands on becomes a data frame in Ganymede, saved in our data lake, and is ready to go for writing some scripts on top of or building a business logic pipeline on top of. That can be lab instruments, that can be apps, that whether it's scientific specific apps like ELNs and LIMS, or generic apps like Excel or analysis apps. That can be existing pipelines, informatics pipelines. We can integrate those, as well as integrating your entire cloud. We have plenty of cases where we integrate with S3 buckets, with Google Cloud, Azure, et cetera. So with that, I'll hand it off to Nathan B. to run through a demo. And Nathan, I can just let me know when you want to click through these slides, and I'll hand it over to you when you open up the app.
Great. Okay, so before we get to our demo, I just want to create a little bit of background here. We're going to be in this demo taking a look at some bioreactor data, analyzing it, and then throwing it over into a system of record. So first of all, if you're not familiar with bioreactors, they're basically ways to grow like living organisms. And so they typically feed in a medium that the organism is going to grow in. They have a variety of sensors, and then you take those sensors, use that to measure what is happening within the bioreactor. So in the lab right now, a lot of these are on-prem OPC connectors that are connected to data historians. You can get the raw data out and do the analysis, but there's a lot of limitations to the amount of context and metadata that you're actually collecting. So although you may have the raw data, you're not going to know exactly what was happening in the with that experiment. And so that's one of the things that Ganymede really excels at, is making it so that you can connect that metadata to the raw results. We can move on. Great. So for today's example, we're going to be looking at a 48 well microbioreactor. So this is a little bit different than the diagram that I showed before. Instead of having one big tank, this is essentially 48 little teeny tanks that are all controlled and measured. So this one happens to be a BioLector Pro. It's made by M2P Labs, which has been bought by Beckham Coulter, and it measures the biomass in the individual well, the pH, and the dissolved oxygen. So we're going to take a look at some data that looks at that over time, so that you can do some analysis on the various growth mediums that you could put in there. So we're going to take this raw data, and I apologize for this slide. It's a little bit hard to see, but it might actually show some of the complexity of the raw data there. In this particular output from the BioLector Pro, there's approximately 2,500 lines of metadata that needs to be parsed, and then there's a comma or a semicolon delimited table that has a variety of data that it's keeping track of. So that includes things like the dissolved oxygen pH, but it also keeps track of when a shaker starts or what the sort of user inputs are. So there's a whole bunch of data that you're collecting that's much further beyond just the basic analysis that we want to keep track of. So we're going to take that raw data, we're going to analyze it, find some inflection points in the curve. So for instance, this is the BS3, so the backscatter. And we're going to look for some inflection points there, so just some basic analysis. And then we're going to output the results to a system of record. For this particular demo, we're going to be using Coda, which is a notebook, an online notebook service that is similar to Notion, if you might be familiar with that. We're going to use that as an ELN proxy. It is, not something that we necessarily recommend that you use, but shows our ability to basically interface with anything that has an API. So with that, we will go to the window. And let's share a different window.
Infinite Hall of Mirrors.
Yeah, there we go. Okay, there we go. So this is a very boring screen that shows the sign-in to Ganymede. Ganymede allows you to use any SSO that you want to, so Microsoft, Google, Okta, and you can just sign in. So this is our demo tenant here. So I'm going to sign in, and each customer gets a private secure tenant that they can use. In this case, we're, again, using the demo tenant, and I'll just kind of go over a little bit about what this screen is and how you would use it. So this is what you see when you first log in, and this is where the scientists would go in order to access any one of these processes. So on the left, you can see we have a variety of navigation. I'll go into each one of these individually. At the top, we've got our documentation, some notifications, and some admin settings. But the meat of what we're talking about here are these flows. So a flow is essentially a pipeline that allows you to take some raw data, do some analysis on it, and output it somewhere. And so we're going to be taking a look at this bioreactor demo v2 flow. And what you should be able to see here is the flow view, which is essentially where you interact with this flow. So this flow takes 3 inputs, CSV and 2 Excels, and then runs the processes, and then you can see what is happening below it. So this is the other screen that the scientists would go into. So to kind of start this off, how do you get files into here? Well, the first and easiest way is just to drag and drop. We're going to go create a new version because we've seen this file before. We also have any number of different other ways of getting files into Ganymede. So we can do things like a Windows connector, or we can read it directly off of an S3 drive. We can also do something like a file watcher, where we take a look at an individual file folder. I should have just opened that. An individual file folder, throw a file into it, and have it automatically upload here. So that allows you to essentially have any number of different connections to get your files up onto Ganymede. So let me just go ahead and put these 3 files here, and we're going to click run. And that's it. Demo over, I guess, because this is all that the scientists would have to do. You can see down at the bottom here, we can see that this flow is running right now, so it's doing the processing, but that's all the scientists would ever need to really interact with. Just kidding about the demo. We'll keep going here a little bit about what's actually happening in the background. So the first thing that we're going to take a look at is what happened to those files. As Nathan was saying, everything in Ganymede is very sticky, so everything is saved. So if we take a look here, and we can take a look at bioreactor, for instance, file, we can see that that file was actually saved into our data lake, and we can see, for instance, the details of exactly what flows it was run in. So this allows you to have full traceability of all of your files and a full record of everything that was put into Ganymede. So once we have these files in there, what's actually happening in the background? Well, in order to explain that, we're going to go to the flow editor and take a look at bioreactor v2. And you can see here, essentially, a series of what we call nodes that are doing individual processing on the files that we just inputted. So each node here, so for instance, bioreactor file, takes an input and has some sort of usually tabular output, and does some processing in the middle. These are generally what we call low code. So they allow you to just drag and drop if you want to. So for instance, if we wanted to create a new node, we have a huge variety of them. But if I wanted to just create a new Airtable import, I could edit it, make some modifications here in order to know which Airtable table I was actually trying to reference, and then just hit done, save it, and it would just work. However, a lot of the time, we don't necessarily want to just, or there needs to be some sort of interaction with the code. So part of the paradigm that we have here is that everything should be editable. So if, for instance, we click on this edit button, we're going to be put over into a Jupyter notebook, which we allow basically web modification of the code, so that you can change anything that you want to about how this node functions. So in this one, we're doing a query to select some data from the data lake. We're doing some analysis here, and then some kind of complex analysis. And then we are outputting that into a data frame so that it can be used later in the flow. And so this is all of this data. If you run it and then click on this run this particular cell, will save everything that you've just written. So all of this function code, all of the query code, into the Git repository that Nathan referenced earlier. So everything is always versioned, everything is always saved, so that you can go back and take a look at other versions of the flows, other versions of your data, other versions of your code. So this is kind of a complex analysis here. So how did I actually develop this without just, trial and error with everything? Well, to do that, I used what we have here as our analysis notebooks. So our analysis notebooks are just hosted Jupyter notebooks that allow you to do a variety of, basically to, let's see this continue running here, to do your analysis, to do troubleshooting, to do or just kind of develop your processes in a way that's not interacting with the flow. So we can take a look at the code here. We can see that we just downloaded some queries. This is where I actually developed all of the analysis that we just did. And then just to kind of, to have some sense of what we were taking a look at, we saw that time series analysis for a variety of wells. We have all the wells here, and then we have some inflection points that were calculated so that we could actually pull some points of interest out of this. So with the analysis notebook done, we're going to take a look at what is actually being outputted by this flow. So if we turn on table view, and I apologize, this gets a little busy, we can see that each one of these nodes actually outputs a table. So in this case, we're looking at the raw bioreactor data. Here we can see the inflection points that were calculated by this particular node, and then the medium composition, for instance, that we read out of that file. So if we click on any one of these, we can actually query the data in our data explorer, and you can take a look at it, in a more thorough manner. You can do joins, you can do whatever you want to in SQL, and then you could output this data to a CSV so that you could do analysis in other formats. So now that we've taken a look at that and cross our fingers that everything ran properly, we can take a look at our flow runs, which is a record of everything that has run through the various flows. So this one, the bioreactor demo v2, we just ran it. We can see that it was a success. It had these inputs here and outputted these files, or this file, and then to output to these particular locations in Coda. So we can take a look and see exactly what happened here with our logs. So it'll just take a second for these to load. And then you can see each one of these nodes is green. So we had success for everything. And then we can take a look at the actual logs of everything that happened, so that you can debug or see exactly what that node happened to do. So now that this is actually done, we can take a look at some of the outputs. We have this presentation file, which was a PowerPoint that happened to output for this particular flow. You can see we have a huge list of them because I've run this flow several times, but again, everything is saved, so we always have a record of it. And if we take a look at this, I apologize for my design skills, but you can see that we have some of these various outputs here. So a heat map of the glucose volume per well, for instance. But not only that, if we take a look back at the flow editor, we wrote to a PowerPoint slide, but we also wrote out to CODA. And again, this is something we're using as an ELN proxy. But if I go to CODA now, and I look at bioreactor demo and click on this one, we can see that this is very similar to what you might see in an ELN. We have, the ability to edit this notebook, but then we have things like some of the run metadata. It was actually significantly larger than this, but I sort of truncated it down for this particular table. We can take a look at the actual results of the run. So in this case, we're comparing well A4 with A7. We can see that there were slightly different substrate compositions, and that caused a difference in the growth. So for instance, here we have the biomass, and we could see that A7 happened to grow a little bit faster, but for not quite as much. We can again take a look at the dissolved oxygen and the pH, and everything you have here is saved both in Ganymede and now in your system of record. So to just kind of recap, we took that raw data, we processed it down from its kind of weird format into sane data tables, took all of that data, did some basic analysis on it, and then put it into various systems of record. We can, this is an example, but either end of that can basically be however you want that configured. So if you want to read this in from an API, we're capable of doing that. If you want to output it to an Excel file, because that's the standard that you have right now, we can definitely do that. Or output it to your whatever system of record you're looking for, Benchling, whatever ELN or LIMS that you're taking a look at. So with that, we're going to conclude the demo and send it back to Nathan.
All right. Thanks, Nathan. That was an awesome demo. And I'll just wrap up here with some final thoughts, bookending this a little bit. But so as you saw from what Nathan was presenting, we have a very broad set of capabilities. And what we're really trying to do is say we can connect to anything. And then the data platform that you get out of that is highly effective because everything's there, both reading the data in and saving a copy, but also being able to read it, push it back out and push it where it needs to go. And when Ganymede engages with people, that's our philosophy as well. Alan here on our team, our founding scientist, is here and helps drive us thinking from a very scientific perspective from the get-go. If you want to build a great data lake, and if you want to have a great home for all of your data, part of it is having the infrastructure to power that and building that. But then the other part of it is, what are you capturing in that, and how do you drive adoption? And one of the things that makes Ganymede very effective for our clients is that we start by understanding the science and the actual bench workflow very deeply. You have to. If you want to incentivize scientists to adopt these new tools, you have to demonstrate some value to them. It's not enough to just save their data and to have it. You also have to drive some automation and make their lives better. And when you make their lives better, you accelerate them, and that ultimately drives time to market. And those sorts of operational gains, in our opinion, honestly, significantly outweigh the gains of abstract data analysis on the overall data set that you've created. Operational insights are everything before you can get to the point of saying, oh, I'm going to do some sort of automated machine learning based DOE or anything like that. You just have to have the data. And so we start by scoping out the process and schema. We deeply understand how things like ELNs are set up or MES systems are set up. We interview the scientists and work with them to understand their process very deeply, so that when we integrate an instrument and we integrate an ELN, we're not just saying, oh, we're going to dump some instrument data into the ELN. We're saying we're going to take the instrument data, we'll automate what the scientist was doing with their analysis. They don't have to manually move files around anymore. They don't have to do the same repetitive curve fit anymore. They can have a human in the loop review step if they want, but they shouldn't have to do the repetitive parts. They shouldn't have to click around through 10 different apps and move files between them. It should just flow. You should press the button on the instrument and your work should be done as much as it can. And so that requires understanding what they do very deeply. So phase 2, when we build the integrations, because of our infrastructure, we build integrations tremendously quickly. We build integrations in a matter of weeks at peak, and with our clients, we'll churn through dozens of integrations for instruments, for apps, for the analytical tools, and bring everything online very quickly and do it in a way that's very satisfying for the scientists and help drive adoption. And when you drive adoption, that also creates better data coverage for management on the end to go in and look at dashboards and use BI tools to drive those higher level operational insights from the data that's been captured. And this is a profound change for companies ultimately, getting everything online and having data in a single place where you can actually reason about it and see it and touch it instead of having to go into physical computers or go into scattered apps and file stores. It's something that across the board improves metrics significantly. But the key metric that ultimately we're all working towards is improving time to market, improving time to IND or time to clinical trials. And when you have all your data in one place, and if something goes wrong, you can just go with a couple clicks or with a single query, go figure out, oh, where did this data come from? Which instrument ran it? I need to go figure out, oh, maybe this instrument's out of calibration. That closes the loop so much faster and can radically reduce the cycle time of experimental cycles from R&D up to later stage process development and clinical work. And so that has a radical, very material reduction on time to market. But a lot of that's enabled by removing data entry errors as well through complete integration, making it so that when you operationalize processes and bring them online for the first time, the developer time to do that is very fast. If you change your science, you should just be able to go in to a cloud-based code editing environment and make that one-liner change that you need. You shouldn't have to go talk to your engineering team or work through some deploy pipeline. You should see what the impact will be, have a good version environment to do it, but then be able to do it very quickly. And that makes our integrations updatable and buildable very quickly. And then finally, having management have this universal visibility to everything from the specific granular low-level scientific data up to higher level descriptions of inter-team or inter-process dependencies where they can see the throughput in a single place across all the places it's stored, ELNs, MES systems, QMS, inventory management. That's also a very powerful tool for management to be able to debug the process as a whole. And this drives cost savings too, whether it's the fact that you're saving bench scientists time and not needing to hire as many bench scientists ultimately, in terms of increasing asset utilization and making it so that you don't have to spend hours futzing around on the instrument computer when Ganymede does the data processing for you. So now you're out of the way and the next scientist can come and start using the instrument. And then finally, our infrastructure is very optimized for this kind of scientific data. And so it makes it, in many cases, we find actually more cost effective than clients building things raw on things like AWS, because we've already selected and curated efficient best of breed technologies here. So with that, we'll move on to Q&A. But something I want to note definitely is appreciate everyone sitting through the webinar so far. Ganymede ultimately, for myself and my co-founder Benson, is a moonshot to try and create a more automated and more online software-enabled biotech and manufacturing industry. And so, although this webinar is a very high-level description of some of our philosophy and a demo, we'll be also trying to go through in future webinars and future material that we produce, brain dumping as much as we can of what we observe, because as much as we're building Ganymede as a tool for everyone, we also want to share any insights we can gain from a product perspective for the industry so that we can generally help accelerate best practices in terms of cloud and software enablement here. So very much looking forward to that. There's a couple questions that are already submitted in the chat, so let me answer those before we jump into general Q&A. So we had a question around how do we ensure data protection for client data? And that's a great question. Something that undergirds everything that we do is our engineering team, and generally how we approach the world is very cloud infrastructure first. Our head of engineering is from Alphabet and worked on some of their cloud infrastructure in a box architecture there. So very much an inspiration for Ganymede as well. And so we take the most modern approach we can with infrastructure everywhere, which helps ensure security. So everything, of course, is always fully encrypted over the web, HTTPS, everything like that. But also, we do periodic pen testing. We're working towards SOC compliance. We'll have that soon. But at a deeper level as well, the way that we work with infrastructure is hyper modern, I would say, where we spin up infrastructure dynamically with the press of a button. Everything that we do is using infrastructure as code and fully deployable per client and controllable per client. So we have what we call single tenancy, where every single client gets their own completely separate infrastructure stack. That isolates your data even more, makes it so that there's no chance of intermingling data between people. This, as much as you want to encrypt and protect data, there's also risk always that at the product layer, you could have data leaking across 10 different clients' environments unless you're fully separating the infrastructure stack. So that's something that we believe in, is that if we're going to be good at offering infrastructure to people, we also have to actually offer them their own infrastructure. And that's a big point for us in our mind. So I would say we have a very modern security approach and are constantly auditing and monitoring and using best practices on the encryption and security side. But at the same time, we also take that to a deeper level in the product itself. There was a question on monitoring Azure and S3 buckets. That's absolutely something that is very easy for us. It's a couple clicks to set something like that up in Ganymede. We can monitor them in the sense that we can take a file when it appears in them. You can fully mount them into Ganymede and sync out all the data from them into Ganymede if you'd like. We're very flexible there. And we will directly, for S3, IAM into it so that we're not actually running over the API necessarily, or a public web API in the traditional sense. Do we have a pipeline for instrument data to go from instrument to agent to ELN without requiring a scientist select and upload a file? Something like automated folder monitoring and processing. Yes, exactly. So when we install our agents, we can configure that behavior. And by default, what we'll do is say every time a file appears, we can do something with it and we'll run it through some automation. You may want to actually pause and have the scientist either annotate it or tag it or kind of review what some of the analysis being done is. So we offer that mode as well. But there's plenty of cases where it's literally scientist presses a button, file is created, Ganymede sees that, pulls it up to the cloud, analyzes it, puts it into the ELN or LIM system or MES. Or same with things like OPC, we'll be sitting somewhere on the network or on the physical device and checking OPC tags periodically and then doing something with it. It can be fully automated. And there was another question on managing clients' data and whether the data leaves the client's environment. Do we keep a copy of the original data, or does Ganymede own and manage the parse data? So hopefully some of that was answered with what I mentioned about the infrastructure approach, but from sort of a data management approach. So everything stays within Ganymede unless you want it to be exported to other systems. And Ganymede can delete copies of either its data or the original source data as people like. By default, we always like to say you should just keep data everywhere and not delete data if you can avoid it. But certainly for certain use cases, especially as people think about like CSV, CSA, sometimes it can be seen as advantageous to delete original copies of the data so that you have only one source of truth copy of the data, or vice versa. Say, okay, I'm only going to keep the original data here, not keep a temporary copy elsewhere, and only use that, do analysis and then render some results elsewhere. So by default, we save all data everywhere. We'll leave a copy on the original instrument. We'll keep a copy in Ganymede. We will also push it where it needs to go, but we can also selectively delete either the Ganymede or the original instrument data if that's desirable for people. Do we own or manage the parse data? We manage it for you, but it's yours. Everything created in Ganymede, the code, the raw data, any sort of business logic within it is all owned by you. And we see ourselves as cloud infrastructure. So it's the same as if you built something on S3 or AWS generally. Anything you build there is yours effectively. The one thing that we'd like to build is as we build instrument connectors, we'll have that available for anyone. And so instrument connectors created in Ganymede can be shared across the ecosystem. And that's part of our strategy, is to ultimately make instrument connectivity more ubiquitous. If the system is single tenant, how do upgrades work? So we have central control over this. So we periodically will upversion just in a continuous CI/CD way the systems, and we'll roll out those updates to each of the individual tenants and whatever their stack is internally. So we have a fair amount of control over this, and everything is automatic updates, unless you specifically need to remove automatic updates for some reason, like our GXP capabilities. And then there's a question about how about pushing things like work lists to local clients? Absolutely. So Ganymede is, what I like to say is, we are human time, we're not machine time. We've optimized our pipelines to run, I would say, in single-digit seconds, which is great. Like if you press the button on the instrument, it'll run. It completed in the time you saw Nathan Ballou demoing it, and then it'll push it where it needs to go. So it feels like, okay, I press my button and then the data is in my ELN. We don't necessarily run millisecond level control over like SCADA systems, but what we can do is absolutely write data to anything we see. So yeah, we can definitely, with our Windows agents, we can push work lists into automation control systems. We can also interface and integrate with schedulers for lab automation. And so that's how we think about the sort of automation world. All right, and then I see also a couple of questions in the chat, so let me answer those. So going through these, any requirement for how data is deposited, for example, in Benchling to make processing work? No, you can write whatever you want into Benchling. You can write, so what we do is we expose anything integrated, including things like Benchling, are just exposed as a tabular data interface. You just write a record and it's done. And we handle the actual injection into the system. So really no requirements. It will have to match if there's constraints set in Benchling, like if Benchling has limitations on what in your schemas of what has been set up and how it accepts the data, it will have to match. But actually writing it is very easy. And we can write in anything, whether it's data, we can write in images, we can write in files, etc. Is Ganymede able to handle native instrument files or only flat file exports? We handle everything. So this is actually, we handle flat files, we handle native instrument files, certainly things like FCS files, those sorts of more semantic files. And one thing that we have is we can even, in a sense, handle files that are encrypted and not parsable, or binary files that require opening things up in a manufacturer software to export. Because what we do is our Windows agents are able to control software like that and automatically trigger export. So if you have something, for instance, like, not to pick on Miltenyi, but max quant flow cytometers are one of the most frustrating things for people when they go try to integrate them, because they're very physically locked down. It's very hard to get into the OS. And they also emit, although they emit FCS files if you set them, they also emit these MQD files, these Miltenyi max quantify data files. And those can't just be opened. There's no parsing library for them. You need to run the max quantify software. So we can either control that on a Windows PC or actually spin up a VM as part of the Ganymede pipeline. We can have pipeline steps that are actually running as an analysis app of some sort to just take that file, automatically export it back to something like an FCS file or a flat file, and then continue running with it. So we have pretty extensive capabilities to help with those sorts of cases. And we can even run, as part of those pipeline steps, an entire app. So for instance, flow cytometry analysis apps, everyone always wants to do their own gating. And scientists want to do that manually. So we can run those in, similar to how our Jupyter notebooks just open up as a tab from Ganymede, we can run entire analysis apps in Ganymede's cloud environment and just open them up as a tab for you to do something interactively and then continue. And then finally, how about CFR compliance? Yeah, so CFR, GXP kind of capabilities we're working towards, and we'll have more there by the end of the year. We have some clients that we've committed to moving towards that with. it should not be too much of a problem. And we've done some initial exploration here, because one of the advantages of Ganymede's approach is by saving everything all the time forever, all the data, when it comes to something specifically like the CFR requirements, that data retention is very easy. And it's not just saying, oh, there are audit logs that can see what happened. You can literally see structured data with traceability back to everything from the get-go. And so that's certainly something that we're pretty confident about and are working towards with clients and happy to talk further on. And then for broader GXP, things like CSA kind of requirements, that's also where Ganymede's infrastructure, because it's all completely software defined and all the code is visible everywhere, it's very transparent and easy to go see and audit what is built in Ganymede and what sorts of code is run and test it. That's all very native to our environment. So working through things like CSA is a breeze when it's not having to go figure out, okay, well, do I need to go audit how this program is constructed? Do I need to audit this spreadsheet and its logic and all these things? It's much more condensed into saying, okay, we expose all the logic that drives everything in Ganymede, and you can just see where it is and go validate it or work on it. Yes, will we have access to a recording? Absolutely. Cool. Any final questions? All right, well, I will turn it back to Alan, but always feel free to reach out to us. We're available, of course, on things like LinkedIn. Our emails are just our first names at ganymede.bio. So we look forward to hearing any questions. And I wouldn't be here if I didn't love chatting about this. So I feel like I've probably given presentations like this 1,000 times literally at this point and never gets old. So thank you all for attending.
Yeah, thank you to both of our Nathans. And I never get tired of hearing it. So always enjoy it. Always have something new to add. I do have some of the poll results, so I was able to find those, and also just dropped a new and final poll into the sort of poll function just to give us your views on sort of how the webinar went, and et cetera. In terms of the overall sort of broader economic conditions, it does seem like that'll be kind of impacting people's decisions, and they're going to be looking to find the right software solutions to really weather this storm. The biotech market did sort of bottom out earlier last year, and there was a lot of hope going into 2023 that things would greatly improve, but I think the broader economy is kind of weighing on that bounce back. And in terms of the other poll question, in terms of what data and integration is most key to you, there seems to be a pretty even split between connecting instruments to the cloud, integrating with apps like ELNs and LIMS, and applying automation to lab data with sort of less of a focus on making the data accessible for analysis, which is interesting. And compliance is really not top of mind, which I'm sure the FDA will be very happy to hear. I think with that, I just want to thank all of you who joined. And for those of you who couldn't, we'll be sharing the recording of this webinar via email. It'll be up on our site very shortly. If you enjoyed the presentation and you do think that Ganymede could be useful for you and your team, or you just want to learn more, feel free to reach out via our website or directly to one of us, including myself, via email. We can schedule a quick call. I'll also point out that we'll be at SLAS in San Diego next week, so please come join us at booth 245. And then we're also going to be at the Lab of the Future conference in Boston on March 9th, and that Nathan will be delivering a keynote address there. If you didn't catch all of that, no worries. I'll be sharing these details in the follow-up email. I just wanted to sincerely thank everyone again. Hope you enjoyed.
Thank you.
Take care.